Hello, this is Javier Carretero Morillo and Sagar God. We are part of the Learning Meat System and Control class by Dr. Marcia Sesclon. Our title is Optimization of Anesthetic and Hemodynamic Drug Delivery Problem in Simulated Patient Environment. Next slide, please. Okay, so here we have the simulator. And this is the Pkpd patient model. It's model to try and mimic the hemodynamic system of a real patient. So basically, during this simulator, we have drugs. The drugs are here as seen is the propofol, remifentanil, atricorium, dopamine, and the sodium nitroprusside, the SMP, which are inputted into a patient. And then the Pkpd models are then creating the outputs, which we have the bispectral index, the Ramsey agitation score, the cardiac output, the mean arterial pressure, and the neuromuscular blockage. Here, the propofol mainly affects the BIS, or the main spectral index. Remifentanil mainly affects the Ramsey agitation score. Dopamine affects the cardiac output. Sodium nitroprusside affects the MAP, and the atricorium, the neuromuscular blockage. Our goal here is to try to regulate the drugs to then keep the outputs within the given range, which is shown on the right side, the 40-60%, -4 to 4, 65 to 110 for both the cardiac output and the mean arterial pressure, and then for the neuromuscular blockage, 0 to 100%. Then during this simulator, we have a disturbance. And this disturbance is to try to mimic what a patient would basically feel or go to throughout a surgery. Because during a surgery, you have different stimulus going on. First, we have the intubation, where the doctor literally opens like the throat and puts a tube inside so the person can breathe. Next, we have the doctors go and then they try to get some test results just to know if the patient is like allergic to some drugs or not, and many test results. And then later on, we have the opening, like the actual procedure where they open, then they do whatever they have to do inside your body. That's like the actual surgery. And then lastly, they close up your skin or everything. and lastly, they just put you in a room and wait for your body to heal. And then this disturbances, whenever they open or you have a disturbance, they make all the BIS and all the outputs drop or go all over the place. Therefore, that's why we're using drugs to try to make them within the given range. Next slide, please. Okay, so here we have it's kind of like the same model as before, but this is like how it looks like in the simulator. So on the left side, we have the inputs. Then we have I guess the big image, which is like the main thing, the main simulator. And then on the right side, we have the outputs. And this is based on the UNESCO simulator.
Open source simulator, yeah.
There you go, open source simulator. Next slide, please. Okay, so here is the original UNESCO simulator with all the subsystems and everything. However, next slide, please. On the next slide, here we just rearranged it to make it look a little bit neater. And then we also just included some loops, some error loops, and all that. But okay, let me explain what's going on here. So at the very top, there we have what I explained earlier within the whole surgery, which is a disturbance. So we have the disturbance, which only goes into the BIS. For some reason, they only focus on the BIS for the disturbance. Next, we have the propofol, which propofol, as you can see, it goes into different things. But the very top one, it goes into the BIS again. And then we have the remifentanil, which for the remifentanil, we have two different outputs. One, actually, the remifentanil affects everything. The BIS, the MAP, the CO, and the NMB, and also the RES score.
Right there.
Yeah, there you go. But okay, so back to the BIS subsystem. So there, the very top three little categories are the propofol, which is at the very top, And then we have one that enters with propofol and remifentanil together, which that one, whenever you inject both the propofol and the remifentanil into your body is thinking that it's like a new drug. So you have like different metabolic rates within your body. Therefore, we have like a whole different category. And then next we have the remifentanil just by itself. Okay, and then the very last output right there for the BIS is dopamine, which for that one it was unexplained. Therefore, when we built this, we just didn't take that into account just because we didn't really understand it. Okay, so next we have the dopamine. Dopamine is affecting the cardiac output and the mean arterial pressure. And next we have the SMP, which also affects the cardiac output and mean arterial pressure. And for this, we have many more inputs because also the propofol affects both of them as well, and also remifentanil. And at the very last, we have the atricorium, which that one affects the NMB, which the NMB is also affected by
Remifentanil. Yeah.
remifentanil. There you go. Okay, next slide, please. Okay, so how else was the project? So first, it was the implementation of certain blocks in the simulator, which were causing mathematical errors. We just changed a couple of the blocks to make it work properly for our loops and whenever we ran the simulator. For the second point was understanding the drug to output interaction, which the main one was what I explained earlier was the dopamine influence on the BIS. It was unexplained, and we were trying to make understand it and run many trials, but we just decided it was better if we just took it out and go with what we understood and knew and how it ran. Okay, so number 3 was the patient-to-patient variability. So the patient-to-patient variability, we just, during our runs and simulations, we just focus on one patient, because whenever you are at many patients, it gets, it was first it's very time consuming understanding what all of this simulator meant. So we thought the best approach would be just to focus on one patient. But for further research, the patient variability needs to be accounted by using learning techniques such as the MPC, gain scheduling, interactive learning, and other learning techniques. And the last point is the inconsistencies of the input values between different papers. So as we understood this simulator, we read many papers, and then we saw some inconsistencies with some of the constants. Therefore, we just focus on just the main UNESCO paper. Okay, next slide.
Yeah. So I'll start explaining how we made those changes, how we implemented our simulator in according to how we wanted to work, especially after we were not able to understand why those mathematical errors were being caused. So we decided to remodel the simulator in our own way. And so we started with basics. We started with focusing on just the BIS. The Propofol Pkpd model looks something like this, where we have the drug being infused into the bloodstream and being metabolized by the by the muscles compartment and the fat compartment. And so the pharmacokinetic, which is the PK model, and pharmacodynamic, which is the dynamic model. Kinetic model is how the drug actually reaches the muscles and the fats, and the dynamic model is how it is being metabolized. So all those aspects were necessary to be understood in order to execute them, and so we did. We read through different papers, and by the same authors, UNESCO and group, we came up with we saw the similarities between the simulator and the papers, of course, and we just decided to make a new simulator with the same things in order to better understand why those mathematical errors were being caused. So for starters, with just the BIS, we have the PK model that looks something like this, based on different coefficients, based on different compartments. These coefficients are relative to each and every compartment based on their volumes and the quantities being moved into them. We have the PD model that looks, that's right over here. We have the controller from one of the from one of the papers that we tried to execute, and we successfully did. And this is how the control action really works. The drug is infused. The pharmacokinetic model tells us how the drug is traveling through the body to the muscles and the fats. The pharmacodynamic model is the metabolism of all that. The Hill function over here is an equation, a mathematical equation that calculates the BIS, the bispectral index, which tells us the level of anesthesia. We also introduced a disturbance in order to make this more realistic. And and when we're taking a feedback, we also introduce a noise. This negative feedback is taken into account with our reference, which is, let's say for BIS, it was 40 to 60. This reference minus the calculated BIS goes as an input into the controller, which based on this input gives out the Propofol output. And so this control action is executed. In the Simulink, this looks something like this. And you can see I have highlighted the controller and also a filter for measurement of the noise, filter for measurement noise. The Propofol PK and Propofol PD models are over here. The disturbance model is introduced as in the original simulator. Then the BIS is fed back to the reference. And this is the reference, again, UNESCO and team. The two noteworthy things here are, again, as Javier just mentioned, single patient applicability. And we have removed the PADE approximation of the BIS delay, which was causing the solver errors. We tuned the PID manually, trial and error. It was not really difficult, to be honest. We found the range being met in less than 10 seconds. We were able to meet the range between 40 and 60 based on these PID gains. We also tried to use Amigo Tuner, but its execution would require more studies. There's also other PID tuners in MATLAB itself, but we were able to successfully get the values manually for this patient. And so we decided to further our studies based on this. For the experimented control schemes, I'd firstly like to talk about the scheme that we were able to execute, and then the literature review of the learning techniques that need to be done in the future. First of all, we have the PID controller right here for the Propofol infusion. Now, this PID controller, based on the gains that we saw a few slides ago, was sufficient enough to control the BIS in the range. Remifentanil input was steady and and the RAS score was in sufficient range as required as well. When the challenge was tuning a controller for dopamine and SNP inputs. And so firstly, we tried to have a PID controller for both of those. But again, since both of those are multi-active with different components in the simulator, like the cardiac output and the mean arterial pressure, fixing one of the PID controllers would always mess the other one up. So we started to take one PID at a time, and we figured out that using a PID controller for SNP was a very good idea. And at the end, we dropped the PID and only had the P and D aspects of the PID controller. So a PD controller and a tuned PD controller for SNP, PID controller for the Propofol was good for this patient's control applicability. On this slide, I have all the results. Now, as you can see, the BIS, again, from the range of 40 to 60 is well maintained throughout the simulation period. The RAS scores, which needs to be between -5 and +4, is well maintained in the range. The cardiac output, which is different from patient to patient based on their weights and heights and other parameters. For this particular patient, it was required to be maintained between 5.72 to 9.68 liter per minute, and we successfully did execute that as well. The MAP is well maintained between the 65 to 110 millimeters of mercury. And we have the NMB score well maintained between 0 to 100. Now, this is also because of a drawback of the simulator that they do not possess a good pain neuromuscular blockade model, which is why they just, for initial studies, they just expect us to control it from 0 to 100. And we didn't really have to do anything to the etrocurium input other than keeping it within the limits, of course. There are certain other approaches that we came across and we studied. We did try to execute them as well. This is a PID cascaded approach where we have such a model if you can see, there's an infusion of the drug. The PK model gives an output. This concentration goes into the PD model. The PD output is then taken into account with the target PD output that goes into the first PID controller, which controls a target concentration. Now, this target concentration is fed back as a positive feedback, taking the pharmacokinetic first concentration as negative, and this error goes into the first PID, and this PID ultimately controls the infusion of propofol. And this is how the execution in Simulink looks like. We have the Pkpd model, the Hill propofol function, which converts this data into the BIS value. Now, we also have the disturbance model, of course. This BIS value is taken in as feedback, and this BIS error is then inverted using an inverse Hill function, which is the inverse of this function in order to get the concentration, which is then fed back into this first PID. The concentration is met, and then the second PID, which controls the infusion of the drug. And this is how the cascaded approach really works. Other than that, we also had model predictive control strategies studied, where you can see the inverse Hill function, the inverse Hill sigmoid function was used again. And these strategies are pretty noteworthy as well as research worthy. Each of these strategies could be implemented and published for sure. In this strategy, we have a target BIS input going into the inverse Hill. So we just take the target concentration to the controller this time, which then gives a propofol input to the patient linear model. And then the concentration works and the BIS is taken as the feedback again. Again, with the inverse Hill, we just take the concentration back to the controller. And this is how, this is another method to control the entire system using the concentrations and not the BIS. Again, the Simulink model is presented right over here using inverse Hill functions to get the concentrations, and so on. There were a few other strategies, namely the gain scheduling, of course, for incorporating patient-to-patient variability. And we also came across an extended prediction self-adaptive control scheme, which looks something like this, where BIS is taken into account, and the inverse Hill sigmoid function gives the concentration, which goes into this EPSAC controller. The input goes into the patient model. The BIS is again taken. BIS is taken as feedback along with the inverse function. The concentration is fed back to the controller, and these different strategies could be used and implemented using the patient simulator, and the work can be done in many different with many different strategies, as mentioned before. The conclusion, the future scope. Javier, would you like to?
Sure. Go ahead. I can conclude. So in conclusion, so the simulator was very user-friendly. We had the freedom to do whatever we want with the simulator, run as many trials as we want. We were able to change various different, but it's very user-friendly. Then we use different strategies for the Propofol reference model, the BIS reference model, the compartmental effect model, and the inverse signal Hill model. And for the research-worthy control schemes, Sagar explained earlier, there's the PID and PD control with gain scheduling, the cascaded PID control with gain scheduling, the model predictive control, the EPSAC strategy for model predictive control, the model reference adaptive control, and then we also have the iterative learning, which this will be a really good, I guess, model, really good for iterative learning because you need a repetitive. It's all about repetition, and then for this, it'll be great for iterative learning. Yeah.
Lastly, we just want to acknowledge all the authors that we have read papers from, especially UNESCO and their team for also developing the amazing simulator. And of course, a great many thanks to Dr. Sescon for being a wonderful guide throughout the semester and throughout the duration of this project. And thank you all. Thank you for watching.
Hope you guys enjoyed it.
Like we did. All right. Good day. Bye-bye.